So, hi, I'm Julia. I'm a fourth year, and today I'm going to be giving you a lecture on bacteriology. So if you could please join. So this is a operative room. I'll also post the thing in the chat so I can move on from it. So yes, so start off with bacterial structure and physiology. So that's the first section on your syllabus. And here is the syllabus for that. I'm only going to cover this part, as I think the other parts have probably come up in other places. And I think will also be covered just in the other bits. So here are some of the key features of bacteria. They're generally about 1 micrometer, and you can see how this fits in with other organisms and cells. So bigger than viruses, but smaller than human cells. They are prokaryotes, so some key differences between prokaryotes and eukaryotes. So first, they don't have membrane-bound organelles. They are haploid, so only have a single copy of a chromosome compared to eukaryotes, which may be haploid or diploid. And usually prokaryotes have a single chromosome. Prokaryotes do still have some filaments, but their cytoskeleton is less well developed than eukaryotes. And there are also differences in the translational machinery, so the ribosomes are different. And this is an important difference as it can be targeted by antibiotics. And then also peptidoglycan is an important structural protein that's unique to bacteria. And again, all the differences are important because they're the things that can be targeted specifically to bacterial cells rather than attacking human cells. So some other structures that are found in bacteria. So you have pili, which are structures on the surface of the cell that attach to other cells. You can also get shorter pili, which are called fimbriae, that help bacteria attach to surfaces. So pili are formed by the polymerization of pilin subunits, and they have multiple important roles in pathogenesis. So they can be important for DNA uptake and therefore contribute to genetic exchange. They can confer movement known as twitching motility, which is different to the movement that flagella cause. And they also allow bacteria to form aggregates by pili from different bacteria binding to one another. And they're probably most famous for their role in bacterial adhesion. And one of the best characterized examples of this is the type 1 pili of uropathogenic E. coli, which I'll talk a bit about later. There's also capsule. This protects the cell, helps it retain moisture, and can help with adhesion. And also the capsule can help prevent phagocytosis. And then there's the flagella, which is long and whip-like and aids in motility of the organism. So the flagella, as you can see in the diagram here, has 3 key components. So the hook, the basal body, which is the motor, and then the filament. And this can allow bacteria to move within the host. For example, through chemotaxis, bacteria can sense presence of food and swim towards it, or can alternatively swim away from toxic molecules. The flagella also contributes to movement, for example, in ascending infections in the urinary tract, where bacteria can use the flagella to go against the flow of urines and cause ascending infections going up into the kidneys. And flagellin is recognized by TLR5, so the host's immune system can respond to it. And then there's also plasmids, which are circular DNA structures and can contain resistance genes, but more about those later as well. So bacteria are generally 1 micrometer. Of course, there's some variation in this. And in bacterial identification, it's important to look at the bacteria and the shape of it. So the most common you'll come across are the coccus, which is spherical. So for example, Staph aureus, or the bacillus, which is also a rod, like E. coli. And then here are some others as well that you may come across. And then bacteria also have characteristic cell arrangements. So some will grow as chains. So for example, strep, or some growing like clumps or clusters like grapes, like Staphylococcus species do this. And then, rods tend to grow in chains. And so when identifying bacteria, this is the first thing you'll look at to try and figure out what it is. Bacteria also have very characteristic cell envelopes. So gram staining, as you probably know, is one of the key tests used in bacterial identification. But to understand how that works, it's important to know the difference between gram-negative and gram-positives. So both types have a cytoplasmic membrane and a layer of peptidoglycan. However, the peptidoglycan cell wall is very thick for gram-positive organisms, and there's no outer membrane on top of this layer. Whereas for gram-negative, they have a thinner peptidoglycan layer, but then an additional outer membrane too. And another key difference is that teichoic acids are present in gram-positive bacteria, but not gram-negative. And gram-negative have LPS, which gram-positives don't have. But it's also remember that some bacteria are gram-indeterminate. So the outer membrane, which is only present in the gram-negatives, has symmetric lipid distribution. So the inner leaflet is phospholipid while the outer leaflet is LPS. And the outer membrane forms a permeability barrier against agents such as bile and antimicrobials. So gram-negatives are inherently more resistant to antimicrobials. And the outer membrane is also a target for insertion of the membrane attack complex and bacterial lysis by complement. Molecules on the bacteria surface also determine the tropism of the bacteria, so the cells it binds to and enters, or where the bacteria survives in the body. So for a bit more on peptidoglycan. So this is a molecule that is only found in bacterial cell walls, and it provides rigid support and helps maintain the cell shape. There's some details about the structure of it there. So it's a polymer of 1-to-4 linked N-acetylglucosamine and N-acetylmuramic acid. And then peptides from adjacent strands are often cross-linked. And humans have 2 nods, so the receptors that can detect peptidoglycan, so nod 1 and nod 2, and they can detect different forms. So gram-positives and gram-negatives tend to have slightly different forms of peptidoglycan. And the peptidoglycan fragments containing MDAP, which are mostly from gram-negatives, are detected by nod 1, while nod 2 detects fragments that are common to peptidoglycan from gram-positives or gram-negatives. So another important molecule is LPS. So it's composed of 3 parts. So it has the lipid A, which acts as an endotoxin, the core polysaccharides, and then the O antigen, which is a repeating polysaccharide chain of varying length. Lipid A is the domain that's integrated into the membrane, and the O antigen sticks out. Lipid A is endotoxic and can lead to a systemic inflammatory response syndrome, which is SIRS. And this includes fever, hypertension, disseminated intravascular coagulation, and activation of complement and macrophages. So, for example, in meningococcal disease, the levels of LPS correlate with the disease severity. And in fact, it's actually the mass inflammatory response provoked by the LPS that is responsible for the damage and the pathology of the disease. So the LPS binds to TLR4, which leads to inflammatory cytokine production, and inside cells, it can bind to caspase 4, which leads to cell death. And just with O antigen, so this is also involved in virulence and pathogenesis, and it can have some bacteria express polysaccharides which mimic host molecules, and this can have different effects. So it can induce local immunity, which can then damage cell barriers through antibody-mediated mechanisms and increase invasion, or alternatively, it can prevent antibody production by acting as a camouflage, so aid in immune escape. And it can also bind to host lectins, or in some cases has been linked with autoimmune disease such as Guillain-Barré, which can follow infection with Campylobacter jejuni that expressed LPS, which is a mimic of the GM1 ganglioside. So this is just a table summing up some of the key features that are different between gram-positives and gram-negatives. So the main things are gram-positive has a thick multi-layer of peptidoglycan and has teichoic acids, whereas gram-negatives have a thin peptidoglycan layer and have an outer membrane and have LPS, the endotoxin. So then for some things on bacterial variation. So this can be intergenomic or intragenomic. So intergenomic is when genes are acquired from another bacterial cell's genome, and this can be done by horizontal gene transfer. Or there's also intragenomic variation, which is when the bacteria makes changes in its own genome. And this can be done by point mutation, which has similar mechanisms in eukaryotic DNA. And also by phase variation, which is a heritable yet reversible gene expression and is a change in quantity of genes, whereas antigenic variation is a change in the gene expressed, so a qualitative change. Phase variation is the variation of protein expression within different parts of a bacterial population. So there are a few different, so a gene can be switched on or off, or can be expressed to a different amount in certain cells. There are a few different ways it can be mediated. So it can be by invertible elements. So for example, the type 1 fimbriae in E. coli Fim-A major subunit. So by inverting the region of DNA that encodes this, it can determine whether the protein's expression is turned on or off. And then another way of mediating this can be by sequence repeats. So for example, port A in M. meningitis, changing the length of the repeated nucleotide sequence can determine whether there is reduced transcription in the amount of port A expressed, or there is a complete stop in the translation. And then, so antigenic variation, I'm sure you'll know a bit more about this, as it's common with viruses too, and also parasites, I guess. So it's an alteration of the protein or carbohydrate on the surface, so it's a qualitative change. And it's used for a mechanism of immune evasion for bacteria. So, for example, where in phase variation, the amount of porine that was expressed by N. meningitis was decreased or stopped being transcribed completely, in antigenic variation, you'd see a mutation that would cause a change in the pore that was expressed. And as opposed to viruses, which have the constantly mutating genome, antigenically variant bacterial pathogens often have evolved mechanisms that use a stable genome, as this aids in protecting the fitness of the progeny, so they can sometimes like switch between which antigen they're expressing. So, for example, with Neisseria bacteria, they do this with the type 4 pilus, which is an important colonization factor that also plays a role in the pathogenesis of the disease. So then for intergenomic bacterial variation, this slide sums up the main mechanisms used to transfer DNA between cells. So transformation is uptake of naked DNA. Transduction is using bacteriophages to package DNA and spread DNA between bacteria. So bacteriophages are viruses or bacteria. And then conjugation, which requires physical contact, and then exchange of genetic material via a mating pilus. So this tends to be how plasmids are swapped between cells or transferred to cells. So some more of those elements. So plasmids can replicate independently of bacterial chromosomes, and they can carry resistance genes and be transferred between cells, either of the same or different species, by conjugation. And the plasmids can be large, encoding the DNA required for the conjugation, or they can be smaller and not encode this information. Transposons are genes that are transferred between or within larger pieces of DNA and typically consist of 3 genes. A transposase responsible for the excision and reintegration of the transposon, and then a repressor gene and the drug resistance gene. And this will be flanked on both sides by DNA sequences that facilitate the interaction of the transposon with larger DNA, which can be a chromosome or a plasmid. And then integrons are mobile gene cassettes that provide the mechanism for new genes to be added to the chromosome, as well as the machinery to ensure that the new genes are expressed in the bacteria. And then bacteriophages, as I said, are viruses that infect bacteria, and they can also cause the spread of resistant genes between bacteria. So now for a couple of Socrative questions. So first, which endotoxin is found in the outer membrane of gram-negative bacteria? Okay. Yes, so you all got that right. Maybe it's an easier question. So yeah, it's LPS. And then, oh sorry, I'll move it on. And then what's it called when bacteria uptake naked DNA? Okay, amazing. So yeah, most of you got this too. So it's transformation. This is naked DNA. Transduction would be with the bacteriophages, conjugation for the plasmids when there's physical contact, and then point mutation is a method of intragenomic variation. So next, for the treatment of bacterial infection. So again, that's your syllabus. And then I thought first, people always forget about the physical methods used to deal with bacterial infection, which are also important, as you need to control the source of infection. Otherwise, antibiotics are unlikely to work sometimes. So especially with abscesses, surgical drainage is required, because otherwise the antibiotics probably won't reach the abscess and you'll still have infection. And then debridement is the medical removal of dead, damaged or infected tissue to improve the healing potential of the remaining healthy tissue. So this is often done for things like necrotizing fasciitis or skin infections. And then on to antibiotics. Oh no, sorry, some key definitions first. So antimicrobial interferes with growth and reproduction of a microbe. Antibacterials are things that harm bacteria. So they are all antibacterials are antimicrobials. And then there's 2 important types of antibacterials. So bactericidal kills the bacteria, whereas bacteriostatic just halts the growth of the bacteria. So this diagram is quite busy, but it does cover all the main antibiotics I think that you need to know and their mechanisms. So starting with beta-lactams, these are far more effective for gram-positive bacteria, as in gram-negative bacteria, the peptidoglycan is protected by the outer membrane. The enzymes involved in peptidoglycan synthesis are called penicillin binding proteins, so PBPs, and these are the primary target of the beta-lactam antibiotics, which are structural analogues of the peptidoglycan. Glycopeptides also target the cell wall, but they actually target the peptidoglycan itself. So, for example, vancomycin binds D-alanine peptide, the crosslinks in peptidoglycan, and resistance to glycopeptides actually comes from bacteria using a different amino acid, so using D-lactate instead of alanine. Sulfonamides and trimethoprim both block different enzymes in the synthesis of folate. So sulfonamide inhibits DAS, whereas trimethoprim inhibits dihydrofolate reductase. And these are often used together for maximum impact. Then for inhibitors of nucleic acid synthesis, we have rifampicin, which inhibits RNA synthesis, and this is used as an anti-tuberculosis drug. Then quinolones and fluoroquinolones inhibit DNA gyrase, which is an enzyme that helps with the unwinding of double-stranded DNA, and therefore these prevent DNA replication. And then metronidazole is often used for anaerobic bacteria, as this antibiotic contains a nitro group that's chemically reduced by bacterial ferredoxin, and this produces a reducing agent in the cell, which leads to damage in vital pathways. And something to think about is there's synergism between antibiotics. So if two antibiotics have different mechanisms, then they can sometimes be used together synergistically, so enhancing the effect of the other. So for example, you could use a cell wall and a protein synthesis combo, and often bactericidal and static antibiotic will be combined. And this synergism from the start of antibiotic therapy can sometimes help to reduce the emergence of resistance. So then I think also important to cover the mechanisms of antibiotic resistance. So what stops all of these working? The first thing is decreased influx of the antibiotic. This is especially used by gram-negative bacteria, and antibiotics that are hydrophilic, such as the beta-lactams and tetracyclines, are particularly affected as they are often reliant on porins to be able to cross outer membrane of bacteria. So the bacteria can change the porins, and then this will stop the bacteria entering. So an example of this is that there was an experiment looking at clinical isolates of Klebsiella pneumonia collected before and after antimicrobial therapy, and the isolate collected after therapy showed shift to expression of a small porin, and this correlated with decreased susceptibility for a number of different beta-lactam antibiotics. Another important mechanism is increased efflux, so pumping the antibiotic out of the cell once it's got in, using membrane pumps. An example of this is the TET efflux pump for tetracycline that causes resistance to tetracycline, Another mechanism is drug inactivation. This can happen by a number of mechanisms. So a drug can be modified by acetylation, phosphorylation, or adenylation, and the aminoglycoside enzymes, so that AMEs, are an example of this, and they covalently modify aminoglycoside, inactivating it. Alternatively, the drug can be destroyed. So beta-lactamases use this mechanism. They cleave the beta-lactam ring present in penicillins and cephalosporins. And then, so that's drug inactivation. Then target modification. So this can involve the target being protected. So example is a protein TET-M produced by some bacteria that interacts with the bacterial ribosome and dislodges tetracycline from its binding site on the ribosome. So then the ribosome can carry on with its work. Then the target site can also be modified by point mutations in the gene encoding the target. So for example, MEK-A is present in MRSA, and it's a gene that encodes an alternative penicillin binding protein called PBP2A, which has a low affinity for beta-lactams, meaning that beta-lactams are essentially useless against MRSA. And then another example is ERN genes that encode an enzyme that methylates the ribosome in such a way that it causes multi-drug resistance to macrolides, lincosamides, and also streptogramin B. And final mechanism is target amplification. This is overproducing the target of the antibiotic. So this can be via a mutation in the DNA promoter region for the gene of the target. And this can overwhelm the ability of the antibiotic to inhibit the metabolic pathway that the target is involved in. So as I said before, often antibiotic resistance is acquired via genetic mechanisms, so the plasmids, integrons, and transferons or bacteriophages. And it's important to note antibiotic resistance can also be developed on a non-genetic basis. So bacteria that can be walled off in an abscess cavity so the antibiotic can't reach them, or bacteria can be in a latent state that makes them insensitive to certain antibiotics. So, for example, antibiotics that affect the cell wall wouldn't be effective against a latent bacteria. And biofilms can also impact the effectiveness of antibiotics. And biofilms often form on sterilized surfaces, so for example, like prosthetic joints or a catheter, and these can be really hard to get rid of. Yes, so antibiotic resistance is becoming an increasing problem, and especially in hospital-acquired infections. This could be due to doctors prescribing multiple antibiotics at once, or overly long courses, or just using them where they're not actually really needed. And the use of antibiotics in animal feed, where about 10 times more antibiotics are used in agriculture than medicine, in order to prevent infections and promote faster growth of the livestock. But this could also be increasing the prevalence of antibiotic resistance, especially as horizontal gene transfer can lead to resistance being transferred between different species. And so this is just a diagram summing up some of the methods used in antibiotic stewardship. So, for example, controlling the infection source, only prescribing antibiotics when they're really needed, in the correct doses, and also for a shorter duration as possible. Reassessing treatment, so using maybe empiric antibiotics to start off, but then narrowing this down once you know exactly what the infection is and using the antibiotic that will be most appropriate for this infection. And also just good hygiene and educating people about antibiotic resistance and how it emerges. So now for a couple more sort questions on antibiotics. Let's move on. So yeah, which of these antibiotic classes interferes with the synthesis of the bacterial cell wall? Okay, yeah, so the correct answer is cephalosporins. So yeah, these are, I didn't mention them, I guess, along with penicillins, these also inhibit the cell wall synthesis, and this is what the other ones do. So aminoglycosides inhibit protein synthesis, as do tetracyclines, and then fluoroquinolones interfere with nucleic acid synthesis. And then for the next, yeah, what's the mechanism by which MRSA is resistant to most beta-lactams? Okay, yes, so more of a mixed response on this one. So the correct answer is it encodes your alternative target protein. So it has the MEK gene, which encodes PBP2A, which is an alternative penicillin binding protein. So remember, those are the enzymes that produce the peptidoglycan. And that's what the PBPs are, what beta-lactams usually target. So this alternative form doesn't have as high an affinity for beta-lactam, so they're less effective against it. Okay, moving on to bacterial diagnosis. I'm not going to go into as much detail on this, as I think it's covered in detail throughout your lab books and in practicals. But if you haven't had as many practicals this year and feel you need more help, I'm happy to try if you want to email me. So this is a bad photo, I'm really sorry, but I just took it from my old lab book, so the Principles of Pathology lab book, and I think it's actually just a really useful diagram and worth just learning for MCQs, as then you'll know what they're getting at when they tell you test results. Probably one of the most important tests to understand is gram staining. So yeah, we discussed the differences between gram positives and gram negatives. So how does gram staining actually work? So the first stain you use is the crystal violet, and this binds to peptidoglycan, colouring cells purple. So all the bacteria initially stain violet. And then you add iodine and potassium iodide, and this forms a complex. And then you add alcohol and acetone. This is used to decolorize the cells. And as gram negatives have much less peptidoglycan in this walls, this step essentially makes them colourless, while only some colour will be removed from gram positives. And the alcohol also dehydrates the thick gram positive cell wall, so the stain iodine complex is trapped inside. And then a counter stain is applied. This is usually safranin, but sometimes fuchsin, and this colours the bacteria pink. But because the gram positives are already purple, you can't see the pink, whereas the gram negatives will appear pink. So the end result is that gram negatives are pink, whereas the gram-positives are the violet colour. And again, remember that some bacteria, such as TB, are gram-indeterminate. So some other tests, so McCunkie agar. So this has a pH indicator, neutral red, which is when there's acidification, colonies will be pink or red, whereas when there's alkalization, this will produce colourless colonies, so still be the red, brown, like dark red colour that the agar normally is. And the agar also contains lactose. So lactose fermenting organisms will produce acid, so these will grow as pink, whereas non-lactose fermenting organisms will grow colourless. And then also the agar contains bile salts, which select for gut-adapted bacteria and inhibit the growth of most gram-positive cocci, sorry. And then, yeah, this is just a quick reminder of what the different types of hemolysis look like. So alpha, beta, and yeah, the non-complex agar. And here are some of the other tests. I'm not going to go through the details of how to do them, just sort of quickly what they're meant to show. So the catalase test can be used to differentiate staphylococci and streptococci, and it tests the ability of bacteria to break down hydrogen peroxide. And if it's positive, it indicates staph, whereas if it's negative, it indicates strep. And the coagulase test can be used to identify Staph aureus. So it's an agglutination test, and if there is agglutination of beads, then this is positive, and that indicates Staph aureus. So then there's Lansfield antigen testing, which is used to differentiate beta hemolytic streptococci based on the carbohydrates that they express on their surface. And the oxidase test, which tests for presence of cytochrome oxidase. So if bacteria contain this enzyme, the originally colourless reagent will turn to an oxidized coloured product. So, and then a list of some of the oxidase positive bacteria you may come across. Okay, so hopefully, oh, okay, I showed you the answer. I'll pretend to go back. So yeah, what colour would gram-positive bacteria be? Amazing, everyone got that right. And then the next one, even though bacterium was catalase and coagulase positive, what is it likely to be? Okay, great. So it's Staph aureus. So the strep species would be catalase negative, and then Staph epidermidis would be catalase positive but coagulase negative. So Staph aureus is the one that would be catalase and coagulase positive. So on to some of the pathogenesis of bacteria. So again, there's your syllabus. So you need to know about the Koch postulate. So the original Koch postulates were used to define a pathogen. So he used these methods to isolate Bacillus anthracis and also TB. So the requirements were that the microorganism was isolated from all suffering diseased animals, and this microorganism could then be grown in pure culture and then injected into a healthy animal, and the disease would be reproduced, and then the microorganism could be recovered again from pure culture, proving that it was that microorganism that was causing the disease. And the molecular Koch postulates are how pathogenicity determinants are identified. So a virulence factor should be expressed by a pathogen, and then if you delete that gene, it should lead to attenuation of the infection in a relevant model. And if you complement that gene, so put it back in, I guess, then that should restore the virulence, so you know that it was deletion of the gene that caused that effect. So here are some definitions. So the virulence is the degree of damage that's inflicted by a pathogen. Infective dose is the number of microorganisms needed to cause a disease. Bacteremia is bacteria in the bloodstream, but this doesn't necessarily isn't associated with symptoms. So activities of daily life can cause bacteremia, which isn't normally associated with symptoms. But septicemia, which isn't really used anymore, but that's blood poisoning. So that's when there's bacteria in the blood and it's causing symptoms, I guess. And then septic shock is when you have sepsis with hypertension that can't be corrected by fluid resuscitation. So the recognition of bacteria surface structures will trigger immune responses. So PAMPs are pathogen-associated molecular patterns, and then these are recognized by the host's pattern recognition receptors, so PRRs. And these receptors can either be membrane-associated, such as the toll-like receptors, so TLRs, or they can be cytosolic, such as the nod-like receptors or the RIG1. So as I said, so an example of a pili that is pathogenic is the type 1 pilus of UPEC, so uropathogenic E. coli. So UPEC causes urinary tract infections, and it has two important types of pili. So the type 1, which binds in the bladder, and the P. pili, which binds molecules further up the urinary tract. And the tip adhesins of both of these contain lectin domains. And lectin binds to oligomannose. So, yeah. So this is just a quick diagram, shows how the infection occurs and progresses. So the bladder's luminal surface is lined by umbrella cells, so these can expand as the bladder distends. And the umbrella cells form an asymmetric unit membrane, which form plaques covering the surface of the urothelium. And these so the AUM plaques contain uroplakins, which are monocylated. So they have these, I guess, oligomannose. And then the type 1 pilus can bind to these monocylated uroplakins. And I guess it's important to understand the molecules they're binding to and how these interactions work, as this can help inform design of anti-infectives. So if you interfered with that interaction, then you could stop the UPEC binding and prevent the infection from progressing. Another important factor in pathogenesis is secretion systems. So there are 7 different types that have been identified, and type 7 is found in Mycobacteria and Staphylococcus. And the others are also found in a variety of gram-positive and negatives. So the secretion systems transport proteins across the outer membrane or through the intracellular membrane and outer membrane. To focus on one of them, just as an example, so there's a type 3 secretion system, and this is found in gram-negative bacteria and is thought to have a common evolutionary origin with the flagellum. 7 families of type 3 secretion system machinery have been identified, and they're made up of up to 25 proteins, but only 9 of these proteins are conserved across all 7 families, so there is quite significant variation between the different families. The genes for these secretion systems are often encoded on pathogenicity islands or located on plasmids, and are currently commonly subject to horizontal gene transfer. So examples of organisms that have these are Salmonella typhimurium and Yersinia. So as can be seen on this diagram, there are a series of basal rings that span the bacterial outer and inner membrane of the gram-negative, and then there is a hollow needle that's connected to this, or a filament in Salmonella. And this structure is then tipped with a translocation pore that's inserted into the plasma membrane of the target cell. And then there's an ATPase that's associated with the base in the bacterial cytoplasm that provides energy for the transport of the proteins. So you think about it, the secretion system essentially transports proteins across 3 membranes from the bacterial cytosol into the host cytosol. And here's just a table showing the diverse roles that a type 3 secretion system can play in different bacteria, from invasion of the host cell to playing an anti-phagocytic role. It can also play roles in attachment or down-regulating host immune defenses. So another important factor that causes the pathogenesis of bacteria is toxins. So toxins can actually be entirely responsible for pathogenesis. You can reproduce disease only by injection of a toxin, and a mutant lacking the toxin is entirely attenuated. And also we can use antitoxins as a vaccine or therapy. So for example, the tetanus vaccine, the toxin is what the vaccine is, and botulinum is a toxin that is used as a therapy, so that's Botox. There's different types, so you can have exotoxins, which are secreted toxins, or endotoxins, which are cell associated, so for example, the LPS of the gram-negatives. And toxins have many different modes of actions, so you get superantigens, which trigger a huge inflammatory response that causes the symptoms of infection. Bacteria can produce enzymes to promote their survival or spread. The toxins can damage or destroy cells. These are known as hemolysins. But don't let this trick you into thinking only red blood cells are affected. The cell membranes can be damaged by enzymes or by the insertion of pore-forming molecules that destroy the integrity of the cell. So both staph and strep produce pore-forming toxins. And then there's the toxins can alter host cells' metabolic machinery, and this is often AB toxins that do this. So AB toxins have 2 subunits. A is the active component, while B is a binding component that's needed to bind to the host cell. So it targets the host cell, I guess. And then when the binding of the B subunit occurs, the A subunit, or the whole toxin receptor complex, is taken into the cell by endocytosis, and then the A subunit becomes activated. Examples of these are diphtheria toxin and cholera toxin. Diphtheria toxin blocks protein synthesis, so the A subunit inactivates transfer of amino acids from tRNA to the polypeptide chain during translation, and B cholera toxin causes massive loss of water from intestinal epithelial cells. And yeah, also toxins can affect nerve to muscle transmission. So example of these are tetanus and botulinum toxins. So just for examples of toxin-mediated disease, so this is cholera. So the cholera toxin has a distinct structure. So it's a 1A, 5B. It has 5B subunits that are responsible for engaging with the host cell. So these determine the tropismal niche of the cholera bacteria. And then the toxin can enter the cell, where the A subunit of the toxin ribosylates G protein, which activates the G protein, which stimulates adenylate cyclase. This leads to the formation of more CAMP, which causes the CFTR channel in the luminal membrane to open, which leads to chloride leaking out of the cell. And then water follows the chloride ions out of the cell. And that's why cholera causes a watery diarrhea and leads to dehydration. Superantigens, they cause non-specific activation of T cells. So normally T cells, of course, would need a specific antigen to be activated, but this isn't the case for superantigens. They can just activate the T cell. So this leads to polyclonal T cell activation, and far more T cells than usual are activated, and this leads to massive cytokine release. Although the aim, of course, of this cytokine release is to kill the bacteria and protect the host, the huge amount of them causes damage to the host tissues and cells, causing symptoms of infection. And some examples of this are TSS, so toxic shock syndrome toxin 1, which is an exotoxin of Staph aureus, which causes toxic shock syndrome, and also enterotoxins, which are a staphylococcal toxin that cause gastroenteritis, so that's food poisoning. So another example is clostridial toxins. So these are produced by Clostridia bacteria, such as C. difficile, which is a gram-positive rod that's spore-forming. And this bacteria causes diarrhea, inflamed colonic mucosa, and pseudomembranous colitis. So C. difficile has two exotoxins, so toxin A and toxin B. Toxin A is one of the largest known bacterial toxins, and it modifies host cell GTPase proteins by glycosylation, leading to cytopathic effects and changes in cellular activities, including damaging the integrity of the cell walls, changing the mucosal secretions, and causing increased chlorine loss. And toxin B is thought to provoke the opening of tight junctions of intestinal epithelial cells, making the cells more permeable and inducing hemorrhage. And yeah, both toxins are glycosyl transferases. I think that's an important thing to remember. Endotoxic shock is septic shock due to the release of endotoxins, so LPS, by the gram-negative bacteria. So LPS is released during reproduction and destruction of bacteria. And the endotoxins then activate macrophages, B lymphocytes, and also lead to cytokine production. And this can cause vasodilation, increased capillary permeability, and activation of the complement and coagulation cascade, so causing all the problems of shock, so the hypertension, and disseminated intravascular coagulation. So in comparison to those mechanisms by extracellular bacteria, there's intracellular pathogens, which are those that enter host cells and are able to survive and replicate there. So this slide shows a summary of the different strategies that intracellular pathogens can use to evade the immune system and the defense of the cell. So there's different methods. So for example, Listeria and Shigella, it's almost as soon as they enter the cell by endocytosis, will escape from the endosome or phagosome and then survive and replicate in the host cytosol. There's difference to Salmonella and Mycobacteria, which when they are endocytosed, survive inside the phagosome but prevent the fusion of the phagosome with the lysosome, so therefore preventing the degradation of the bacteria, whereas other species such as Coxiella and Legionella actually survive inside the phagolysosome, so they've developed mechanisms to survive there. Some examples. So Salmonella is one that survives and grows in phagosomes. So Salmonella has three major antigens: the H antigen, which is flagella, the O antigen, and then the capsular antigen. And the different species of Salmonella are serotyped according to these three different antigens. And also Salmonella has two of the type 3 secretion systems we were talking about before. So one of these is used for the entry of host cells, and another helps with intracellular replication. Shigella is another intracellular bacteria that's also a gram-negative rod, and it rapidly escapes from the phagosome when it's endocytosed and undergoes replication in the cytoplasm of the cell. So Shigella enters the cell using machinery that's encoded on the entry region of the Shigella large virulence plasmid, and this region encodes the type 3 secretion system needed for entry, along with transcription factors and also effectors and chaperones. As Shigella is gram-negative, it has LPS in the membrane, so this binds to caspase 4 inside cells, leading to pyroptosis of the host cell. So pyroptosis is inflammatory cell death, and when the cell dies, the bacterium will be released from the cell and then can go on to infect other host cells. The peptidoglycan in the cell membrane binds to nodes, which then leads to the formation of an inflammasome, which I'm sure you'll hear more about in immunity lectures. And also important to Shigella is the leading cause of diarrhea worldwide. So there's 3 serogroups and 1 serotype. So serogroup A, B, and C, and then serogroup D only has the 1 serotype, so that's S on I. And groups A to C are physiologically similar, whereas S on I is a bit different and has different biochemical metabolism. So the S. flexneri, so the serogroup B, is the most frequently isolated species worldwide, accounting for 60% of cases in the developing world, whereas S on I is more common in the developed world, so it causes 77% of cases in developed world, but only 15% in the developing world. So that just shows how different pathogens can predominate in different places. Yes, and then the serogroup A is usually the cause of epidemics of dysentery, especially in confined populations such as refugee camps. So it's important that bacteria regulate this, the expression of their virulence genes, and so they can do this by sensing changes in the environment. So, for example, temperature, nutrient availability, osmolarity, or the cell density of the bacteria themselves. So, for example, quorum sensing is the ability of the bacteria to detect and respond to its cell population density by gene regulation. So, here's an example of the induction of bioluminescence in Vibrio fischeri. I don't know how to say that. And so you can see that the bioluminescence is only switched on when the bacterial cells have reached a certain density. And another way bacteria can respond to their environment is by 2-component systems. So the signal can bind to a receptor that then leads to a activation of another component that causes the differential gene expression. And there's an important difference between this program change to sensing the environment and variation in bacteria. So when bacteria are responding to a change in environment, the change is detected, and then the message is transduced to change expression, leading to the altered phenotype. Whereas when we're thinking of variation in bacteria, so for example, the response to antibiotic resistance, there's a stochastic change, so a random change in the bacteria in a mixed population. And then a change in the environment leads to some of the bacteria being fitter, so these are likely to proliferate and then predominate in the colony. So now a couple more Socrative questions. So yeah, which of these is an example of a superantigen? Okay, amazing, you all got this right. Yep, TSST1, so that's the superantigen that causes toxic shock syndrome. And then which of these bacteria replicates in the cytoplasm of the host cell? Okay, yeah, so the answer is Shigella. So both Shigella and Salmonella are intracellular pathogens, so Staph aureus and E. coli are extracellular. But Salmonella survives and replicates inside the phagosome, whereas Shigella escapes from the phagosome into the cytoplasm, and that's where it survives and replicates. Okay, so now for some specific pathogenic bacteria. Your syllabus does give you a list of sort of ones to learn about. I'm not going to cover all of them, just sort of a bit about TB and also hospital-acquired infections. So TB is different from the other bacteria we talked about, as it has a different cell wall structure to either gram-positives or gram-negatives. So its cell wall is made up of mycolic acids, and these are strong hydrophobic molecules that form a lipid shell around the mycobacteria. And it's sort of a waxy cell wall that it forms, and the cell wall makes MTB resistant to desiccation, so drying out, and also makes it resistant to antibiotics that would kill other bacteria. So a really specific antibiotic cocktail-like regimen taken over a number of months is required to treat TB. And the cell wall is also antiphagocytic. So TB is very slow growing and causes chronic infection, so it can lie latent and then be reactivated, causing an active infection again. In the lung, TB can lead to the formation of granulomas. So in a primary infection, so when someone first gets infected, a Ghosn focus will form in the lungs, and then T cells respond to the infection, but the interaction between T cells and macrophages lead to a granuloma forming, with the TB enclosed in the center of the granuloma. And then, like when they're responding, the macrophages confuse, which forms giant cells surrounding the granuloma. And then enclosed within the granuloma, as there's less oxygen and nutrients reaching this area, the material starts to die, so you get caseous necrosis, causing caseous center in the center of the granuloma, and this is quite characteristic of TB. So on hospital-acquired infections. So yeah, as it says in the name, these are infections people get whilst they're in hospital, and they can occur because people in hospital are generally immunocompromised or unwell, so they're more at risk of infection, or it can be due to events that happen during their illness and the provision of the healthcare. So, for example, inserting catheters or surgical procedures or contact from healthcare workers can lead to spread of infections around the hospital. So some common infections that are seen in hospital setting are listed here. So UTIs, respiratory tract infections, gastrointestinal infections, especially C. difficile, wound infections, and also bloodstream infections and sepsis. So also it's important, viruses are also a huge cause of hospital-acquired infections, but I've not included them here as it's a lecture on bacteriology. And antibiotics are an important factor, I guess, in hospital-acquired infections, as they can affect the gut microbiota, leading to increased risk of C. difficile infections, which can be a big problem. And high levels of antibiotic resistance can occur, leading to the spread of antibiotic-resistant organisms, such as MRSA, throughout the hospital. So some ways to prevent these occurring are antibiotic stewardship, hand hygiene, and just hygiene in general, I guess, so a clean environment using sterile instruments. And then also, if a sort of epidemic does occur within the hospital, lots of people are getting the same infection, like doing root cause analysis to see where this has come from and how to stop it. So then just a couple of other specific bacteria. So Strep pneumoniae is a gram-positive coccus that's alpha hemolytic and grows in chains. And this is a commensal in the human respiratory tract, but can also, so it's found in the respiratory tract of healthy people, but it can also cause respiratory tract infections. And most commonly, it is the most common cause of community-acquired pneumonia. So pneumococcal pneumonia causes fever and chills, coughs, difficulty breathing, and also chest pain. And if the infection then spreads to the central nervous system, it can cause meningitis. And there are 2 vaccinations available to prevent strep pneumoniae infection. And so one of these is a conjugate vaccine, whereas the other is a polysaccharide vaccine. So then strep pyogenes, so this is group A strep. So if you remember, the group A comes from the Lancefield classification, so based on the polysaccharides expressed on the streptococcal surface. This is also a gram-positive cocci, grows in chains, but it's beta hemolytic. It's an extracellular pathogen. It's found on skin and mucosal membranes, often in asymptomatic individuals, and it can cause pharyngitis, scarlet fever, necrotizing fasciitis, so that's a sort of flesh-eating bacteria, I guess, that kills the skin, causes quite nasty infections, cellulitis. And it can also cause rheumatic fever, so this is sometimes 2 to 4 weeks following a strep pyogenes throat infection, individuals can develop rheumatic fever. So that's fever, and it also affects multiple organ systems. So carditis is the major cardiac manifestation of this, and you get polyarthritis too. So if you get a question where it's saying someone 2 to 4 weeks after a throat infection started getting a fever and more like their organ systems are involved, that's probably caused by strep pyogenes. And yeah, so some of the important virulence factors of this pathogen are the hyaluronic acid capsule, which is antiphagocytic, the adhesins, hemolysins, so the toxins that kill cells, and then also pyrogenic exotoxins that act as superantigens, so cause a massive inflammatory response. And then Staph aureus, so this is a gram-positive coccus which grows in clumps. Yeah, it's commonly found on human skin and mucosa, but can also cause disease, especially if the skin is broken or via a medical procedure, which I guess is why it's so predominant in hospitals. And it can cause skin and wound infections, abscesses, endocarditis, and also bacteremia when it gets in the blood. And then, yeah, an important thing to note is MRSA, which, as I said before, carries the MEK-A resistance gene encoding the alternative penicillin binding protein. So yes, I think that is all I had to cover. So I would recommend for the bacteria, if for all the ones that are on your list, if you just sort of make some notes on them, so the key factors, like the tests that would be used to identify them, and that can help with MCQs. Thank you for listening. Please leave feedback using the link that's on the bottom of this slide, or I think Jethro just posted in the chat.